Okay, got the laser pointer there. All right, good afternoon, everyone. Thanks for having me. I'm a postdoc currently in Bassler's Lab at Princeton University. And like Jen just said, I'm here to talk to you today about our favorite phage, VP882. So in the Bassler Lab, we study the process of quorum sensing, which is the production, release, accumulation, and detection of small molecules called autoinducers, which allow cells to take a head count of their surroundings and decide when to engage in group behaviors. So at low cell density, on the left here, autoinducers are produced, but they can freely diffuse away and are not detected. And therefore the cells will engage in individual behaviors. But as the density of the population increases, so does the concentration of autoinducers. And at a critical point, the cells will then begin to engage in group behaviors like biofilm formation, light production, virulence factor production, etc. And in our favorite genus, the Vibrios, there are two main quorum sensing systems, the first of which on the left here is the LuxO-OPA-R system. And if you're familiar with any of Bonnie's work, OPA-R is LuxR, is HAP-R, only the species changes for some reason. But that system there on the left, at low cell density, LuxO is phosphorylated, leading to the production of these small RNAs, the QRR sRNAs, which will translationally repress this master high cell density regulator, OPA-R. But at high cell density, autoinducers are detected, leading to the dephosphorylation of LuxO, QRRs are lost, and OPA-R is freely translated and can control group behaviors. And then the second pathway is the VQMAR pathway, in which at high cell density, the autoinducer DPO is sensed by the cytoplasmic quorum sensing receptor VQMA, leading to the production of VQMR, which can also control group behaviors. Now, in my favorite Vibrio, Vibrio parahemolyticus, one of the major groups of behaviors controlled by quorum sensing are surface behaviors, like biofilm formation and swarming motility, and that is through OPA-R and its interactions with a third sensory system called the SCR system. So at low cell density, OPA-R is absent. SCR is produced. SCRA makes a currently unknown molecule called the S signal, which is detected by SCRB, which then binds SCRC, leading to phosphodiesterase activity, a drop in cyclic diGMP, and swarming motility. But at high cell density, OPA-R comes in, turns this whole system off, leading to an increase in cyclic diGMP and biofilm formation. But I told you I was going to talk about a phage today. So how does that come into all this? Well, in 2019, we started working with this phage VP882. And VP882 is a temperate phage, so it can go lysogenic within its host. But why we were interested in VP882 is that it actually encodes a homolog of its host quorum sensing receptor, VQMA, which we call VQMA phage. And VQMA phage can also sense that autoinducer DPO at high cell density. And at a critical point, VQMA phage will trigger lytic induction of phage VP882. And we hypothesize that VP882 is eavesdropping on the density of its host in order to time lytic induction to the maximum possible chance of transmission to a new host. And just as one extra little layer here, we know that OPA-R is also modulating VQMA phage's activity. So this phage seems to be integrating multiple quorum sensing pathways into its lytic induction decision. But when I started in the lab, I came in with a question of how do these quorum sensing pathways impinge on the entire life cycle of VP882, not just lytic induction. So that meant asking about when a phage finds a new host cell and infects and has to make that initial lysis-lysogeny decision, do the quorum sensing pathways also play a role in that process? And to investigate that, I started by infecting a panel of quorum sensing mutants in typical shaking liquid culture the way we always would. And I have 3 mutants here to start. Or 3 strains. I have the wild type strain on the left. In the middle is the LuxO D61E strain, which is a phosphomimic. These cells will always think they're at low cell density. And D61A is a constitutively dephosphorylated mutant, which the cells will always think they're at high cell density. And hopefully it's obvious from this graph that the wild type and the high cell density strains, I saw no formation of lysogens after 24 hours of infection, but in that low cell density lock strain, I saw almost complete conversion of the population to lysogens. So we know that at low cell density, phosphorylated LuxO represses OPA-R, that master high cell density regulator. So I thought it's got to be OPA-R. But I deleted OPA-R and nothing happened. And in fact, the OPA-R mutant with the phosphomimetic D61E mimicked the D61E mutant. telling us that phosphorylated Luxo is epistatic for lysogeny. And so the only thing we know between Luxo and OPA-R are those QRR sRNAs. So naturally we asked, is it the presence of QRR sRNAs at low cell density rather than the absence of OPA-R driving lysogeny? And it is. So I overexpressed QRR2, one of the those QRR sRNAs, and we saw that I could drive the wild type population during infection from about 20% lysogen formation up to like 80% lysogen formation. So it does seem that independent of their activity on OPA-R, the QRRs are driving lysogeny in liquid at low cell density. But I told you that one of the big things quorum sensing regulates in parahemolyticus are surface behaviors. So I wanted to know if now we infected cells living on a solid surface, would this pattern hold? And initially it does, while overall levels of lysogeny are lower during surface infection, the pattern is still there with little to no lysogeny in wild type and high cell density locked populations, but an increase in low cell density locked populations. But here, when I deleted OPA-R, that was actually the highest amount of conversion to lysogens I saw. So unlike in liquid, when you infect on a surface, it seems to be the absence of OPA-R rather than the presence of the QRR sRNAs that is driving lysogeny. So what's going on here? Well, I told you OPA-R represses this surface sensing system, SCR-ABC. So I wondered if it's the de-repression of SCR-ABC at low cell density that leads to lysogeny when cells are infected on a surface. And that's the case. If we delete SCR-ABC across the panel of quorum sensing mutants, we can ablate lysogeny across the board. So whereas it's the QRRs in liquid, it's de-repression of SCR-ABC at low cell density on a surface. Now, SCR-ABC, like I told you, controls the cyclic diGMP pool. So we think the obvious next question was, can we tune the cyclic diGMP pool and then tune lysogeny during infection? So to do that, I started with this SCR-ABC mutant in which SCR-ABC is gone. So cyclic diGMP is constitutively high, and we see very little lysogeny. But by complementing back in SCR-ABC, I can drive down cyclic diGMP and drive up conversion to lysogens in that population. And then on the flip side, our OPA-R mutant, in which cyclic diGMP is naturally low because SCR-ABC is de-repressed, I see increased conversion to lysogeny. But if I overexpress SCRE-E554A, which is a version of SCRC that's locked in its diguanylate cyclase form, thus driving up cyclic diGMP, I can turn lysogeny off, essentially. So by tuning cyclic diGMP down or up, I can tune lysogeny up or down, if that makes sense. And this is great, but we wanted to know if this is specifically due to SCR-ABC or if it's just the global cyclic diGMP pool on a surface that's controlling this. So I moved on to using non-specific phosphodiesterases and diguanylate cyclases, the first of which is our non-specific phosphodiesterase TPDA. And you can see this replicates overexpression of SCR-ABC. Just by driving down the cyclic diGMP pool with any enzyme, we can turn up lysogeny. And on the flip side, using a non-specific diguanylate cyclase, GEF-A, we can again turn down lysogeny. So to just kind of sum that all up here, we see that no matter what the physical context of the cell is, lysogeny always seems to occur when the cells are at low cell density. But the quorum sensing dependent pathway to lysogeny varies based on the physical context of the cell, with the QRR sRNAs driving lysogeny in liquid and SCR-ABC driving lysogeny on a surface. And then we know as those cells go to high cell density, both OPA-R and VQMA phage will control exit from that lysogenic state into lytic induction. And so we can say that the entire life cycle of VP882 seems to be controlled by multiple quorum sensing pathways. So with that, I thank the lab, particularly Bonnie and Justin, who kind of kicked off the VP882 work in 2019, and Ned, who helped us with a lot of the calculations for these experiments, and my funding, the organizers for having me. And with that, I will take any questions. Thank you.
I assume this is not just true in Vibrio. It's true in a lot of phages. So is there any application that you're thinking about as far as treatment for infection?
Yeah, it's hard to say. I don't think about phage therapy a lot because we tend to stray away from lysogenic phages for phage therapy because as much as they kill bacteria, they also like to give bacteria all kinds of fun new toys. But if anything, I know when Justin started on this work, he was initially looking at building kill switches using VQMA phage and just the control region of this phage to see if upon induction of quorum sensing, we could auto kill bacteria. And that's interesting because like I said, quorum sensing does control virulence. So it seems to be a process they need to infect. So why not use it to kill them? But how to deliver that is a whole nother question.
Right, so the battery says I'm gonna shout also.
Okay.
So this is really cool.
Thank you.
So I'm curious about your thoughts on some of the native environments where Vibrio can be found and how this might control kind of population dynamics.
Yeah, no, that's a great question. It's hard to say because VP882 doesn't seem to behave like our typical phages, right? You think of a phage, you add it to the culture, all of a sudden all these cells burst and everything clears. We've never seen that with this phage. Like I can add it and wait, and we'll maybe see a little bit of a death curve, but not much. This is not your typical phage. It's a linear plasmid phage. So its life cycle seems to be a little longer. So my thoughts are that maybe the amount of time it takes for it to go through its genomic rearrangements, the bacteria division might actually outpace that. And so this phage is just kind of quiescently moving through the population. But yeah, it's interesting to think about the dynamics in the environment. I have to think on that a bit more. Yeah.
So from what you just said, am I understanding that you need a high MOI to be able, so, in that case, how frequently does this appear in Vibrio phage, right? Is to say, how common
This mechanism? So initially I'm glad you asked this question. When this paper came out, we only found this phage in like 2 strains. But I'm writing up a paper right now. We dove in to all of NCBI and multiple other phage databases, and I've actually found about 24 versions of this VP882 across multiple species, across multiple environments from like 1990 to 2020. So it does seem like this thing is quite far spread and not just in Vibrio. In multiple species that do make DPO, though DPO is a universal autoinducer, not just Vibrios make it. So it's interesting that this phage seems to be taking advantage of it in multiple species.
Very cool.
Thanks.
So thinking about the QRRs. So do you think either from experiments with this bioinformatics, are they targeting other digestive barriers, or do they directly target something phage themselves to trigger?
Yeah, it's a great question. We have no idea. There was a recent paper that came out of Kai Pappenfort's lab where he looked at the small RNA interactions between the phage and the host in Vibrio cholera. So it's going to be a little different than perihemolyticus. He didn't see much on the phage itself. He saw the phage did have its own small RNA and there seemed to be some crosstalk. But yeah, the real answer I have is I don't know, but we're in the process of doing those experiments to see what the QRRs are actually targeting outside and inside the context of infection.
Any other questions?